Hello everyone, good day. This is Ma'am Honey and today we will be talking about photosynthesis. What is photosynthesis? Plants are autotrophic organisms, meaning they can make their own food. And the food making process in plants is what we called photosynthesis. These autotrophic organisms use light energy and convert the light energy into chemical compound such as starch, which is a polysaccharide carbohydrate that every organism uses to power its metabolism. In the process of photosynthesis, we have here the 6 molecules of carbon dioxide plus 6 molecules of water. With the action of light energy coming from the sun, we have the byproducts sugar such as glucose, the C6H12O6, which are usually stored in starches, and the byproduct oxygen, which is released by plants and utilized by animals. Knowing the requirements and products of photosynthesis, let us proceed to the next question. Where does photosynthesis occur? Photosynthesis takes place in chloroplasts, which have an outer membrane and an inner membrane. Stacks of thylakoids called grana form a third membrane layer. Again, the chloroplast is an organelle where photosynthesis takes place and it contains layers. So the chloroplast contain an outer membrane, an inner membrane, and a third membrane which is composed of stacks of thylakoids called grana, which is in plural form, and granum in singular form. Inside these thylakoids are what we called lumen, the red part here. And these thylakoids are also surrounded by a membrane wherein these membranes are where the chlorophyll pigments are embedded. So these pigments contribute to the absorption of light, or these are responsible for absorbing light particles or photons. Okay, so basically it is responsible for the initial interaction between light and plant materials. Another part of the chlorophyll is the stroma. So the stroma is a liquid filled space surrounding the granum. So those are the parts of the chloroplast where photosynthesis takes place. Knowing the different parts of the chloroplast, next question is how light is absorbed for photosynthesis. Basically, photosynthesis occurs in plant leaves where chloroplasts are found. And these chloroplasts contain the thylakoids, where thylakoids contain or are surrounded with thylakoid membranes, wherein in these thylakoid membranes, pigments such as chlorophyll are embedded. Different kinds of pigments exist, and each has evolved to absorb only certain wavelength of visible light. Pigments reflect or transmit the wavelengths they cannot absorb, making them appear in the corresponding color. So example, the colors of leaves, such as the green color. Chlorophyll A and chlorophyll B are found in higher plants' chloroplasts and will be the focus of the following discussion. How does gas exchange occur in plants? The gas exchange of carbon dioxide, which is used up by plants, and oxygen, which is released by plants, occurs through small, regulated openings called stomata. In singular form, stoma. These stomata play roles in the regulation of gas exchange and water balance. The stomata are typically located on the underside of leaves and help to minimize water loss. Each stoma is flanked by guard cells that regulate the opening and closing of the stomata by swelling or shrinking in response to osmotic changes. Before we proceed to our discussion about the process of photosynthesis, let us know the following terms first. ATP, which stands for adenosine triphosphate, is a molecule that carries energy within cells. ADP, which is adenosine diphosphate, binds with a phosphate group to produce ATP. NADPH, which stands for nicotinamide adenine dinucleotide phosphate, which is a reduced form of NADP positive, is the product of the photosystem 1. PS2, or which stands for photosystem 2, moves hydrogen ions to the stroma for ATP synthesis, while PS1, which stands for photosystem 1, captures the energy to reduce NADP positive into NADPH. CYT, which stands for cytochrome complex, is an enzyme composed of two protein complexes, which are the PQ, or plastoquinone, and the PC, which is the protein plastocyanine. The CYT enables the transfer of protons across the thylakoid membrane, and it also allows the transfer of electron from PS2 to PS1. PQ stands for plastoquinone, is a carrier molecule which is part of the electron transport chain, while PC is a protein plastocyanine which is also part of the electron transport chain. Another is the RUBP, which stands for ribulose biphosphate, in which it reacts with carbon dioxide to form two molecules of 3-PGA. And Rubisco, which stands for ribulose biphosphate carboxylase, is an enzyme which catalyzes the reaction of RUBP and carbon dioxide. PGA, which stands for phosphoglycerate, is the product of the reaction of carbon dioxide and RUBP. And last one, we have the G3P, which stands for the glyceraldehyde 3-phosphate, which is the product of three molecules of PGA plus ATP and NADPH. Knowing all those important terms, now let us proceed to our discussion about photosynthesis. First question is, what are the two parts of photosynthesis? The two parts of photosynthesis are the light-dependent reaction and the light-independent reaction, or Calvin cycle. In the light-dependent reactions, from the word itself light, meaning it uses light energy coming from the sun as well as water in order for the reactions to occur in the thylakoid. These reactions produce ATP and NADPH and release 1/2 molecule of oxygen back into the atmosphere. On the other hand, in the Calvin cycle, or the light-independent reactions, these reactions occur in the stroma and use carbon dioxide and the products of light-dependent reactions, which are the ATP and NADPH, in order to produce the molecule Ga3P. And this molecule will be used in order to produce the sugar molecules, or C6H12O6, which will be stored in a form of starch. The overall function of light-dependent reaction is to convert solar energy into chemical energy in the form of NADPH and ATP. This chemical energy supports the light-independent reactions or the Calvin cycle and fuels the assembly of sugar molecules. Another question is, what are the components involved in light-dependent reactions? The components involved in light-dependent reactions are the photosystem 2, the cytochrome complex, the photosystem 1, the NADP positive reductase, and the ATP synthase. In the photosystem 2, it contains protein antenna where chlorophyll molecules are bound. This photosystem 2 also contains a reaction center where a pair of chlorophyll molecules are found, and these chlorophyll molecules have special property that could undergo oxidation. And when it undergoes oxidation, meaning it loses electron, and these electrons will be transported by the electron transport chain. Part of the electron transport chain is the cytochrome complex, which is composed of the protein plastoquinone and the protein plastocyanin. In the photosystem 1, same with photosystem 2, it contains antenna proteins where chlorophyll molecules are found or bound, as well as the reaction center, which contains this pair of chlorophyll A molecules that could undergo oxidation, which could lose electrons. And another component is the NADP positive reductase. It is an enzyme which is responsible for catalyzing the process of reducing NADP plus or positive to form NADPH. And the last component is the ATP synthase, which is also an enzyme responsible for catalyzing the formation of ATP or the adenosine triphosphate by binding a molecule of ADP and one phosphate group. How does light-dependent reaction occur? First thing that happen in the light-dependent reaction is when the chlorophyll molecules capture the light coming from the sun. And after capturing the light, it will be passed from one molecule to another and absorbed by the special pair of chlorophyll A molecules. And when the energy is absorbed, it will cause the excitation of these molecules until it will oxidize. And when there's oxidation, it loses electron. And this electron will be transported by the electron transport chain, which includes the cytochrome complex, the plastoquinone, and the plastocyanin. The cytochrome complex allows the transfer of electrons from the photosystem 2 to photosystem 1. How will photosystem 2 or P680 replace the missing electron? There is a missing electron since the electron in photosystem 2 is transported to photosystem 1. Missing electron in photosystem 2 is replaced by extracting low energy electron from water. Splitting one molecule of water will produce 1/2 atom of oxygen, 2 hydrogen atoms, and 2 electrons. About 10% of oxygen is used by mitochondria in leaf to support oxidative phosphorylation, and others are released to the atmosphere, while the hydrogen atoms will be released into the thylakoid lumen. Why do electrons lose energy during the transportation process? Electrons lose the energy during the transportation process because the cytochrome complex enables the transfer of protons across the thylakoid membrane. Energy from the electrons will be used in moving hydrogen ions from the stromal side of the membrane into the thylakoid lumen. These hydrogen ions from the stroma, as well as the hydrogen ions released by the splitting of water, will be used in ATP synthesis. How will photosystem 1 or P700 gain energy? Since the energy in photosystem 2 is used up by the cytochrome complex in moving hydrogen ions from the stroma into the thylakoid lumen, there's a need for the photosystem 1 to gain energy. And in doing that, the chlorophyll molecules found in the antenna proteins will capture the energy coming from the sun, or light energy will be captured, and this will be passed from one molecules to another until the chlorophyll molecules or the special chlorophyll molecules absorbed this light energy, causing its excitation, and oxidation will occur. And again, once there is oxidation, there's losing of electrons. So these high energy electrons will be produced, providing energy in order for the reduction of NADP positive to form NADPH. The products of light-dependent reactions are NADPH, which is produced by photosystem 1, and ATP, which is produced by ATP synthesis. Now, how does ATP synthesis occur? ATP synthesis occur when there's too much hydrogen ions concentration found in the thylakoid lumen. And when there's high concentration of hydrogen ions, passive transport or passive diffusion occurs. And when there's passive diffusion, meaning it's the movement of molecules from high concentration to low concentration. Now, how energy is produced in this process? Ions or hydrogen ions build up energy because of diffusion and because they all have the same charge. With that, since these hydrogen ions have the same charge, they will repel each other. And to release the energy, they need to rush through any opening, which is the ATP synthase. So the hydrogen ions will pass through the ATP synthase, releasing energy. And that energy will be used to bind a molecule of adenosine diphosphate and one phosphate group to produce an ATP molecule. In the light-dependent reactions, the products are ATP and NADPH, which are used by the Calvin cycle or the light-independent cycle. Now, question is, how does light-independent reaction occur? The light-independent reaction of the Calvin cycle can be organized into three basic stages. These are the fixation, this is fixation, reduction, and regeneration. First stage of light-independent reaction is what we call carbon fixation. In this process, the molecules carbon dioxide, RUBP or the ribulose biphosphate, and the rubisco, which is an enzymes, are involved. Rubisco stands for ribulose biphosphate carboxylase. Now, rubisco catalyzes the reaction between the two molecules, the RUBP and carbon dioxide. In every molecule or in one molecule of carbon dioxide that reacts with one molecule of RUBP, two molecules of 3-PGA are formed. Since in this reaction, three molecules of RUBP react with three molecules of carbon dioxide, the product is composed of six molecules of 3-PGA. This process is called carbon fixation because carbon dioxide is fixed from an inorganic form to organic molecule. Therefore, 3-PGA is an organic molecule. After the carbon fixation, reduction follows, and in the reduction process, it uses the products of light-dependent reactions, ATP, and NADPH, in order to convert the six molecules of 3-PGA into six molecules of GA3P or glyceraldehyde 3-phosphate. It is considered as a reduction reaction. It is because it involves the gaining of electrons by 3-PGA. Recall that reduction is the gain of an electron by an atom or molecule. Six molecules of ATP and six molecules of NADPH are used. For ATP, energy is released with the loss of the terminal phosphate group, right? So when a phosphate group is removed, energy will be produced and converting it into six molecules of ADP. For NADPH, both energy and the hydrogen atoms are lost, converting it into NADP positive. Both of these molecules, the NADP positive and hydrogen, as well as the six molecules of ADP, will return to the nearby light-dependent reactions to be reused and re-energized. That is for the reduction process. The next stage of the light-independent reaction is what we called the regeneration of the RUBP. Recall that after the reduction, the products are the six molecules of GA3P. In this process, only one molecule of GA3P will be released back into the cytoplasm, which will be used in forming other compounds such as sugar molecules. So, one molecule of GA3P is equal to 1/2 molecule of glucose. So, there's a need of two molecules of GA3P in order to form one molecule of C6H12O6. Since only one GA3P is released, the remaining five molecules of GA3P will be used up in order to regenerate RUBP by utilizing or using three molecules of ATP. To sum it up, photosynthesis has two parts, the light-dependent reactions and the Calvin cycle or the light-independent reactions. The light-dependent reactions use light energy coming from the sun and water in order to do its or for the reactions to occur, producing ATP and NADPH and releasing oxygen. On the other hand, the Calvin cycle or the light-independent reactions use carbon dioxide as well as the products of light-dependent reactions, ATP and NADPH, in order to produce the product GA3P. Two molecules of Ga3p could make one molecule of glucose or C6H12O6, and these glucose molecules are stored in a form of starch. Thank you for watching this presentation, and I hope that you learned from today's discussion. Bye!